please send to Dr. Sisi. We will be able to get it.
Okay. So, good afternoon, good morning. I don't know if there are some good evening people. This is to welcome you all to this webinar series, which is part of many others. This is the third. And again, this is part of the capacity project that is funded by the European Union, EDCTP. This is a we are part, JCOS is part of the consortium that is coordinated by Coimbra University in Portugal. It includes the National Drug Authority in Uganda, Uganda National Council of Science and Technology, Muhimbili University, and the Joint Clinical Research Center. Today, we are privileged to host Professor Rimas Orentas. Is a Senior Scientific Director, Research and Development Immunotherapy in Militenyi Biotech Inc. He leads groups focused on CAR T cells, NK CARs, and hematopoietic stem cell engineering. He's based in Maryland. And Dr. Orentas also holds an adjunct professorship in the Johns Hopkins Bloomberg School of Public Health. He's a co-founder of Caring Cross, a non-profit implementing low-cost approaches to engineered cell therapies. He has led teams that delivered 6 first-in-human CAR T products. So there is a lot of talent here. And he knows how to move products from the lab to the clinic. Previous positions include Professor of Pediatrics, University of Washington School of Medicine, Director of the Ben Town Center for Childhood Cancer Research in Seattle, Scientific Director of Lentigen, a Militenyi biotech company now, and Associate Scientist, Pediatric Oncology Branch at NCI NIH. And faculty positions at the Medical College of Wisconsin. There are many other things, but I will stop here because his CV is very long. Today, I'm co-moderating this webinar with Dr. Lois Baiga. He's one of the first people to be trained in gene therapy in the US. And she will mainly moderate the next session, which is Q&A. I now hand over to our expert who is online to deliver his very interesting lecture inside the lab, how gene therapy works. But he knows how to take things from the lab to the clinic. Rimas, you're most welcome.
Dr. Sissi, thank you so much for that kind introduction. I think it's been more than 2 years since I've been in Kampala, so that's too long. And I remember first meeting so many of your team, not only there, but also at UC Davis, where we started some early training for cell therapy and cell manufacturing. So greetings to all the team and to you, and thanks for the opportunity to share today. So I'm going to start very high level, just so we're all on the same playing field. Just specifically, what is gene therapy, or what we call genetic engineering? It's the use of genetic material to treat, cure, or prevent a disease. And we do this either ex vivo or in vivo. Ex vivo meaning the gene therapy is carried out by removing cells from the body, genetically modifying them, and returning them to the body. So as the drug is a modified cell, this is also referred to as cell therapy, or in general, cell and gene therapy, CGT, is often used to denote this process. In vivo gene therapy refers to the direct administration of genetic material, usually by means of some vector. The gene therapy we're talking about is non-heritable. This means somatic cells only, not germ cells. That means none of this material is passed on to the subsequent generations. It's only for the therapy of the patient at hand. Cell and gene therapy can also use allogeneic cells, not just cells from the patient, but could be another person. It could be from a bone marrow donor during bone marrow transplant, or it could be cells created from cultured stem cells, such as induced pluripotent stem cells, or iPS. Why do we do this? I think this is the why. We live in a revolutionary time for cell and gene therapy. On the left is Jim Olagere, cured from sickle cell disease by genetic therapy of his stem cells. And there's a famous picture of him at the summit of Mount Kilimanjaro. In the middle is someone who's quite well known throughout the world, Emily Whitehead, who with her parents and researchers at University of Pennsylvania founded the Emily Whitehead Foundation. And she was the first pediatric patient cured from leukemia. That's her in hospital on the left. And she's now a young, healthy young woman on the right. And on the far right is Adam Castillo, London patient. He was cured from HIV during bone marrow transplantation for a malignancy. But in the process of the bone marrow transplant, and because of a unique characteristic of that transplant not to be reinfectable with HIV, CCR5 negative mutation, he's cured. And I show this picture because each one of these individuals are just beautiful people, wonderful people, each made in the image of God. And this is why we do it. We do this to bring cures to patients and to learn how to do it better. Cell and gene therapy also is part of a revolution that's been happening in our understanding of the immune system. The immune system normally is thought of as the way we eliminate bacterial or viral infections. But the immune system also holds the potential to recognize and eliminate cancerous cells. However, each cancer type is different and has to be approached based on its own specific profile, genetic and proteomic. On the left is an example of a chimeric antigen receptor T cell. So this is a T lymphocyte, a white blood cell from the peripheral blood, that has a modification of its genome through a gene vector to express a new protein on its cell surface. And this is that new protein, shown here through the schema. This protein is called chimeric because it's built from 3 different proteins that are present in different cells or different secreted proteins in our bodies normally. So on the extracellular aspect of this T lymphocyte, we have the combining region of an antibody, the heavy and light chain of the variable regions. On the inside, we have a co-stimulatory domain and a CD3 zeta signaling domain that activates the T lymphocyte once this antibody portion binds. So just like a T lymphocyte can attack and kill a virus-infected cell, it can now attack and kill anything that bears the counter receptor to this antibody-like moiety. In this case, if it's CD19, which is a marker on the surface of B cells and especially on B leukemias and lymphomas, we get this interaction. The T cell binds with the tumor cell. And in the middle is a scanning electron micrograph of a bunch of T cells binding to a tumor cell and in the process of killing it. And that's what we're trying to do. This is not, this has become standard thought. Over 10 years ago, Science magazine declared cancer immunotherapy as the scientific breakthrough of the year. And on the cover, we see T cells on the attack. So this is basically what we're after. How do we find these targets? That's the central idea of how we identify what can we use our chimeric antigen receptor modified T cell for. We've had success in the B cell malignancies because the pathologists have done a good job. The different types of malignancies we encounter in the B cell lineage can be pre-B cell or pre-B cell. They can be more like immature B cells, or they can be like activated plasma cells, which each have very different antigen expression profiles. So for example, CD19 is in the pre-B cells, and CD38 and BCMA are exclusively expressed on myeloma B cells. So because we have this natural progression through normal B cell development that's in some sense mirrored in the B cell malignancy, we have many targets we can choose from when trying to design CAR T cell therapy for B cell malignancies. More challenging has been identifying targets on the surface of solid tumors or cancerous cells. We don't have a marker like CD19 on solid tumors or cancers. Now, you might think, hey, I made a chimeric antigen receptor modified T cell that targets CD19. It does eliminate all B cell leukemias, but it also eliminates normal B cells. That turns out to be a survivable side effect. This is not always true for solid tumors. For example, we can't eliminate all the fibroblasts in our body or all the muscle cells in our body. Many years ago, we started to understand that we could do differential gene expression. That is, we can look at genes expressed in normal cells versus genes expressed in tumor cells. And in the upper left, they always show this picture from Laura Wood, who's on faculty now at Hopkins, who was in Bert Vogelstein's lab, and they had the cover of Science in 2007. And in this XY plot, they lined up all the human chromosomes, starting with chromosome 1, kind of like a string, end on end on end. And they mapped all the mutations, in this case, that were present in colorectal carcinoma. And these tall peaks, the z-axis, represent the number of mutations. And as you can imagine, the largest peak was for P53. And as an immunologist, when I looked at this said, hey, this looks very much like the surface of a cell, because to an immunologist, everything looks like the surface of a cell. And as we started to do differential gene expression on solid tumors versus normal tumors, we discovered there are certain overexpressed proteins we can target with chimeric antigen receptor modified T cells. Now, these can be immune adhesion or signaling receptors like B7H3 or CD276. They can be cytokine or growth factor receptors like IL-13R alpha in brain tumors, or EGF, epidermal growth factor receptor mutant 3. Or proteins expressed on sarcomas like fibroblast growth factor receptor 4. We can also target gangliosides. There's no protein there at all. It's just lipid and sugar, but we can make antibodies to them. And there was just another paper published by the Locatelli group out of Rome showing beautiful results with an anti-GD2 chimeric antigen receptor T cell in neuroblastoma. We can target proteins that communicate with the extracellular matrix, like GPC3, mesothelin, small molecules or neurotransmitters, and even the MHC peptide complex, which is normally thought as the domain of the T cell receptor recognition. We still can make an antibody to this complex. Now, there's other ways that we can target these cell surface proteins with antibodies, bispecific antibodies, or other binding moieties like aptamers. But we're going to focus here on gene therapy and how to use it. Now, this wonderful diagram created by the Brenton's group shows the chimeric antigen receptor in close-up with all the different potential domains we can modify to make an even more active chimeric antigen receptor. So in the middle, we have the lipid bilayer. To the left is the extracellular aspect, the binding domain. So the one I talked about earlier is the scFv, the binding domain of an antibody. But basically a cytokine, a nanobody, any type of ligand we can imagine on the surface of a tumor cell, hey, if it has a counter receptor, I can use it to activate a T cell. We then link it to the transmembrane domain through amino acid sequences derived from proteins like CD8 or immunoglobulin. Then we need our co-stimulation molecules, the molecules that activate the T cell to bind, proliferate, and secrete the cytotoxic granules that kill that cell. And because we're adding a new set of genes into that cell when we do gene therapies, we can quote unquote armor. That means we can add other payloads. We can add other proteins that are delivered to that T cell in addition to the chimeric antigen receptor. So we could perhaps create cytokines like interleukin 15 or interleukin 18. We could create other small molecule binders. We could secrete bispecific antibodies. We can give new adhesion receptors. So here's a small list of the different ways in which we've started to armor this genetic payload package as we begin to genetically modify T lymphocytes. When we start to do gene therapy, we have to first choose a vector. A vector is how that nucleic acid that's going to do our therapy is delivered. And we can deliver that therapeutic nucleic acid using a virus-like delivery system, a nucleic acid direct delivery, or we can actually do gene editing or gene modifying, where actually we introduce an enzyme or enzymatic system that alters a gene or edits that gene. Now, which one we choose to use depends on the different characteristics that each one has. For example, if our gene vector is a simple virus particle, such as the adenovirus or adeno-associated virus, we are subject to having a very strong immune response. So basically, we get a one-shot delivery with these interventions. Now, adenovirus, we all have a standing immune system too, so it's not used very often directly because of that immune system's response, although it has a nice gene of interest or payload that we can deliver. The adeno-associated virus is much smaller. It's like a small protein BB, but we can put up to 4.5 kilobases in there. We can put a larger payload, about twice the size, if we use an enveloped virus particle to deliver the gene into the cell, whether it's a retroviral or lentiviral-based delivery system. And if we're using different chemical compositions, like a lipid nanoparticle, which is a liposome that encapsulates either plasmid DNA, message RNA, or circular RNA, we can potentially deliver much larger pieces of therapeutic genetic material. However, the specificity is a challenge, as well as finding a chemical composition that gives you freedom to operate. The lentiviral-based gene vector field is based on HIV. HIV is our first genetic vector. I always consider that nature's small joke, that the bane of many of our existence is actually an exquisitely designed gene delivery machine. So in this diagram, you can see in blue is the envelope. Protruding through the envelope is the envelope protein, which interacts with the CD4 cell and a T lymphocyte in the case of HIV. Inside is the protein capsid, the protein core, and the viral double-stranded RNA genome. So when the lentiviral virus, in this case, binds to its receptor, it internalizes, circularizes, creates an LTR that can now be pasted into the genome, and now it's part of a DNA expression complex. If we engineer it properly, we can use this just to create new mRNAs and create new gene products instead of just creating new viral particles. So it changes from being a virus to a vector when it no longer makes infectious viral particles. This again is another depiction of the HIV. In the middle again is the cartoon. Each of these transmembrane domains with this purple tulip-like structure. This is the trimerized GP160, GP41 plus GP120. And on the right is a colorized electron micrograph showing you this is actually pretty accurate depiction of what the lentiviral particle or virus looks like. The bottom part of the slide is the entire genome, just over 9 kilobases. And the genome is divided up into different gene expression families that are regulated and expressed in different ways during the infectious cycle. In blue are the group antigens. These are the structural proteins that make up the capsid and core. We have the polymerases, especially the integrases and the reverse transcriptases that change the RNA genome into DNA. And we have the envelope protein. This is that structure here that we've been talking about. And we have different regulatory proteins like TAT, VPR, and NEF. Molecular biologists and virologists have been able to identify different parts of this viral genome and then split them up into different plasmids, which different gene expression units, which when put together back into a producer cell, creates a new viral particle, or if it's a one-way delivery, a new viral vector. So in lentiviral manufacturing, we've taken this HIV genome and split it into different parts, 3 or 4 plasmids that are transfected into a quote-unquote producer line that then secretes a new viral vector into the supernatant. And these producer-like manufacturing steps are at least thought of in generations. So we have a first generation lentiviral vector, a second generation, and a third generation, which is the generation we're using now. In the first generation, we define that, we can just take out the envelope plasmid, and we no longer need to use GP160 from HIV. We can use the fascicular stomatitis virus G glycoprotein. It binds many more different cells, so it's no longer CD4 restricted, basically anything with the LDLR receptor. Then the transfer vector is the specific expression cassette. So it's bounded by the long terminal repeats and has the packaging element psi, but that's going to be our genetic payload. And then there was a third plasmid that had all those other proteins on it that weren't the genetic delivery package. They weren't the envelope, but what needed to create the gene delivery cycle. We streamlined these three plasmids into a second generation vector families, and we have only the envelope packaging, and then we minimized into just GAG, POL, TAT, and REV. That's all we needed. When we moved to a third generation vector, we now created 4 plasmids. This REV protein was taken off into a separate plasmid. The second plasmid only has the group antigens and the polymerases. And now we have the transfer vector. It's been somehow changed. So usually the 3' and the 5' long terminal repeats, which I'll remind you are here and here, which are the major promoters for gene expression. Well, this 3' is completely disabled, so that once this vector internalizes, it flips, it's no longer functional. So we call this a self-inactivating vector. And when you have a self-inactivating vector, now you need an internal promoter as well. We don't need TAT anymore because that's the transactivator element for the LTRs, because we're not driving expression from the LTRs. We're using an internal promoter, using a standard mammalian promoter, like elongation factor alpha-1 alpha or PGK, or you could use different hybrid viral promoters. In any case, we're now in the third generation, which has 4 plasmids and requires an internal promoter, and it's highly safe. There's no chance that we can recombine and create another type of virus. Now, when we manufacture these, we split this into an upstream process and a downstream process. Upstream, we take these 4 plasmids that I've just described to you, we mix them with a producer cell line, in this case, the HEK293 cells, into a bioreactor. In this case, here's a 200-liter bioreactor. We harvest that supernatant, and then we process it. We filtrate it to concentrate the material with ultrafiltration and tangential flow filtration. Chromatography is used to capture and purify and concentrate. We do another TFF to put it into formulation buffer. It's then finally filled into a glass vial or a plastic bag, and it gets the final label. So this is the process that we do to create a lentiviral-based gene vector. Now, I highlight this envelope here because we've developed a new vector system in which uses a different envelope, and it's able to infect natural killer cells and gamma delta cells quite readily. It's no longer the VSVG protein, it's the baboon envelope protein, or BAEV. And I mention this because we've put this in front of certain regulators, and they had an assumption we were making a new virus or a new vector. And we took a long time. We had to explain, no, we're just changing one of these plasmids to a different envelope. It's already a pseudotype envelope already. It's the VSVG is not normally part of the original genetic material from which we derive this vector system. So this can be changed out. And we've changed it out into the baboon envelope and get great transduction of NK cells and gamma delta T cells, which was not possible with the VSVG type pseudotype vector. So what are the key drivers keeping CAR T costs high and restricting access? We've spent a lot of time thinking about this, and they can be technical, regulatory, legal, or what I call philosophical. Technically, it's the cost of goods, the cost of manufacturing, the cost of the therapy itself, the hospital stays, and all the other ancillary time and treatments. Regulatory is a super important question. Do we have a regulatory framework that creates or limits access to cell and gene therapy? Some countries, perhaps Southeast Asia, there's very little regulation and hospitals go very, very quickly. I recently visited a country in South America that has super high levels of regulation. Everything has to be done in a super strict clean room. And I think that's over-regulating. And that's going to drive up costs and actually make it less accessible. So I think we have to continually have a relationship with our regulators to interact with them and show that what we're doing is safe, it's effective, and we only need the level of clean room, isolation, and really testing that's required. We don't have to overdo this. We've been doing these cell and gene therapies for quite some time now. Legal, I think we can spend hours talking about patents, whether or not they're blocking patents, whether or not patent enforcement policy is being done fairly. But in some ways, the market is somewhat rigged towards people that put out a lot of blocking patents and don't let the inventions to be properly licensed or practiced. Philosophically is how do we price the cost of cell and gene therapies? As we all know, a cell and gene therapy for sickle cell disease, as approved in the Western market and US EU, is $2 million for the Vertex product, $3 million for the Bluebird product. And that's based on a justification of disability yearly costs that were saved from the healthcare system. So, for example, if a sickle cell disease patient incurs $1.9 million of cost over the lifetime of that disease, that should be the cost of the therapy. Now, of course, that's not what we price antibiotics or heart drugs for. So the question of why this has to be the case for cell and gene therapies, I think, has to be deeply thought about by the field, and whether or not we're actually in a system that can allow us to base our reimbursements on cost within a fair patent and legal system and in a reasonable regulatory environment. Since we're talking about the lab, that was the title of our lecture. We're going to concentrate on technical solutions. At Miltenyi, our technical solution has been the use of this device, the Clinimax Prodigy, that could actually be used for true point of care cell manufacturing. Back in 2021, we showed that using this device, which is shown here in a highly regulated, this would be a pharma-like environment where we're making many, many products from many patients. But we showed here in 2021 that whether it was in Michael Moscan's lab in Moscow, Russia, or Marcos de Lima's lab in Cleveland, Ohio, if we had the same gene vector and the same manufacturing platform, we got identical, highly effective anti-CD19 CAR T-cell products. Again, using the same platform, Dr. Vikram Matthews at Vellore, at the Christian Medical College in Vellore, showed that we could actually create these products at a very reasonable price. So this paper in 2023 in bone marrow transplantation outlines the work that he carried out and the cost pricing that he analyzed within an academic network in India. I always show this slide because I want regulators to think about this. This is our colleague, Dr. Nirav Shah at the Medical College of Wisconsin. And in this facility here, he's treated more than 100 patients. That's many, many lives have been saved. This is not an ultra high, super expensive clean room like the one that I showed you here on the right that's used for pharmaceutical manufacturing. This is an academic medical center with 2 prodigies, sterile hoods. It's a controlled laboratory environment. It's the same environment where they process their bone marrow products. But in this controlled laboratory environment, safely and effectively, we can generate CAR T cells. And we love to show this slide as well. This is Dr. David Britton in Pretoria, and the first patient to receive CAR T cells in South Africa, all of sub-Saharan Africa. And we're just pleased as punch that Nicodemus is doing well. In the middle here is Esther Romosele, who is part of our Miltenyi team that helped launch this project. So how are we doing? Well, the B-cell malignancies have given us consistent targets, as we've been talking about, but it's about a 50% long-term cure rate. So diffuse large B-cell leukemias and the B-acute lymphocytic leukemias, we're doing pretty well, even with monospecific CARs, CD19, CD22, or CD20 specific CARs. A little better, perhaps, with tandem CARs that do 2 targets at the same time, and potentially we're looking at how to target 3 at the same time. Multiple myeloma, we've also done very, very well. The commercial CAR products targeting BCMA, they're validated and they're effective, and they have really nice effects. We're going to improve this even further by adding second protein targets, making tandems that recognize BCMA as well as GPRC5D, or there's a number of other targets you can think about for multiple myeloma. For CLL that undergoes Richter's transformation, not as clear that we have good targets, and we could have a scientific discussion about which ones we want to choose next. ROR1 is one candidate, CD3 is another. I talked to lots of investigators in the field, and they each have their favorites. So how are we going to make the next generation of CD19 or BCMA CAR T-cell products better? What are we doing in the lab? Well, the current work seeks to improve the cell product itself. Is it more durable once we infuse it, or do the T-cells become metabolically exhausted when we infuse them? Are we targeting enough targets at the same time? Is 19 and 20 going to be sufficient? And what about the solid tumors? Is one CAR T cell going to be enough? How do we increase the specificity while decreasing the toxicity? Marcella Maus up in Mass General has knocked out interferon gamma, and in some instances, it's less toxic and equally effective. And we keep trying to look at the best logic gate switches, meaning if I have one signal, do I turn on another, perhaps more general signal, but in a specific physiological or even anatomical location? We can do this by choosing better cells. We choose T stem cell memory cells because they last longer when we infuse them. But there are the alternative cell types like NK cells, gamma delta cells, and NKT cells. By increasing the processing speed, the vein to vein time is decreased. We can also have a younger cell that seems to be more active. Many investigators look at engineering the metabolic signals that a T cell undergoes during activation and perhaps giving pharmacologic rest by using kinase inhibitors or by adding drivers of T cell expansion like JUNB, PRC2, we can get a better T cell product. We can also armor, we've talked about armoring before in that initial diagram, by adding what I call decoy receptors. So we can put a dominant negative TGF beta receptor so the T cell is not inhibited by TGF beta, which is a negative immune signal in many solid tumors. And then there's the decoys for PD-1, which is the off signal FAS or CD200. We also have to think about the disease itself. We have to better understand the mechanisms of CAR T cell resistance. Is there an inherent disease resistance through a stem cell-like signature? Are there acquired mutations in the cell death pathway so that the leukemia or the solid tumor doesn't die once the CAR T cell ligates and delivers its death package? Or has there been antigen loss or antigen variation? Well, we've done a lot of work on antigen loss and antigen variation. So this is the reason why we designed bispecific CAR T cells, to address that antigen loss, antigen downregulation, or cellular dedifferentiation. So the first time we actually had a hint of this was in 2015, when the group led by Andrei Thomas Tikhonenko looked at the CAR T cell data coming out of the University of Pennsylvania and the Children's Hospital. And what they found was that CD19, the very protein that was being targeted by the CAR T cell, can mutate. It can even drop an entire exon so the CAR T cell no longer sees it on the surface. It's similarly in the laboratory, Elad Jacoby, when he was in Terry Fry's lab at the pediatric oncology branch, was looking at strong immune pressure using both mouse models and the clinical samples that came out of the NIH as well as out of Seattle. And there they showed there was a lineage switch that occurs into a more myeloid-like lineage. And in 2018, the same group, along with us, we show that, hey, when we use a CD22 CAR, we actually get a downregulation. So this is not a mutation. This is not an antigen loss, but it's a downregulation. So on the upper right are all the different patients who were analyzed for this publication. In the lower right, I think it's very obvious. So this is a flow cytometric profile of the leukemia prior to treatment. So you can see this strong expression of CD22 on leukemia. After they receive the anti-CD22 CAR T cells, you can see that this expression strongly drifts down. So this is why we started to develop the tandem CAR T cell products. Now this has led to a potential commercial product by Miltenyi, which is a tandem CD19/20 CAR that's moving towards approval. So on the upper right is a cartoon showing its tandem structure. It has both a CD19 and a CD20 binder, along with the BB and CD3 zeta signaling domains. And in many cases, we can give this fresh, 14 days vein to vein time. So leukapheresis is carried out, lymphodepletion with cyclophosphamide and fludarabine, as is per standard for all CAR T therapies, and then given a fresh infusion, and then follow-up time. We're very, very excited by the data that's coming out by this. We're looking at progression-free and overall survivals. This is from last year's ASH meeting. It'll be updated again at this year's ASH meeting as we move towards approval for Xamtocell. And we have very nice persistence of the CAR T cell product in the peripheral blood. So this is the CAR T cells per microliter, and this is over a period of 180 days. This work is primarily being carried out by Dr. Nirav Shah at the Medical College of Wisconsin and David Miklos at Stanford University. So our innovation continues. So in the very top line is a schematic of the Xamtogene product, Xamtocell, and it's the CD20, CD19 binders. Now this is the Lu16 And this is the FMC63 binder. So they're mouse derived, linked to a BB zeta. We now have human versions of these, human binders. So we hope to be able to do a CAR after CAR treatment and have less immunogenicity. We've also created different formats, such as the third generation CAR, which has not just BB, but also CD28 and BB zeta in a tandem format. And we're also testing different hinge domains and different transmembrane domains in this chimeric antigen receptor T cell. Now, you can create two binders in one glycoprotein, which is the top part of this slide, or you can create two separate glycoproteins and express them on the surface of a cell. So when you have this P2A skip element, the ribosome comes along, skips, and then makes a second strand. So you actually get two proteins on the cell surface. And in a bicistronic vector, as this is called, we can have CD20 with the 28 zeta signaling, and then CD19 on a separate chain with BB zeta signaling. Now, the reason why I bring this up is because the tandem space has all of a sudden become very, very crowded. So yes, this is our product, but also now J&J as well as Kite are coming out with their own versions. So the J&J vector is essentially the same as ours, except it has a CD28 transmembrane, and they acquired this from a company called Able Zeta in China. So it wasn't really from it. It's similar to how they acquired their BCMA CAR. And Kite has gone at a bicistronic approach. And so this is a schematic of Kite's vector. Now, it's important when we're creating these chimeric antigen receptor modified T cells for treating leukemias that we treat models that hopefully begin to show you the differences between how effective these different structures can be. So in this system, we implant the Raji lymphoma cell line that expresses luciferase. So when we add the enzymatic substrate for luciferin, we get a emission of a photon, and we can capture that in a CCD camera. And so when a mouse is glowing, that means it has lots of tumor. So when we give tumor alone or untransduced, that means T cells, but no genetic modification. You can see the mice start to glow more and more as the disease grows, and then they have to be sacrificed because the disease burden is too high per the protocol in the different veterinary facilities. So we stage these mice, we divide out the signal equally so there's no bias, and then we give CAR T cells a week later and then do a weekly blood draw and a weekly analysis. And all of those different versions of tandem CAR T cells I just showed you were all pretty effective at controlling disease. So the graph on the lower right is the cumulative light signal from each of these murine subjects. Now you can see that at day 63, we decided to do some, day 56, we did something interesting. We added some fresh tumor. Now this would measure if there's any CAR T cells still around, whether or not they're exhausted, and whether or not they're still active. Interestingly, and I skipped about 20 pages of in vitro data, just skipped right to the in vivo data, our Xanthocell is still pretty good at controlling this tumor. So that's the red triangle. It wasn't actually, there were a couple assays where it wasn't quite as strong as these other formats, but in controlling disease, it's outstanding. And we look at these other formats, we can see that actually switching 28 and BB, or BB and 28, they have very different outcomes depending on which binder is connecting to which signal transduction molecule. So in this case, this open purple square doesn't control the disease as well as the open blue triangle, although they're very, very similar CAR T cell products. So I often say that in these mouse models, as CAR T cells go to die, but you have to pick the right model and you have to do really rigorous tests to start to understand which ones are more active. Now, we've proposed using a trispecific CAR that has a CD2019 and then a P2A skip element and a CD22 element. And the different arrangements of signaling molecules such as OX40, CD20, or BB or Zeta, how these are arranged and how these are going to work, they're each being tested in different clinical centers. The Medical College of Wisconsin, the James Cancer Center, Ohio State, investigators at the University of Maryland School of Medicine are each trying different flavors of these. Now, I wish I could tell you in this presentation which of these work better, but this data is all going to be shared, or at least two of these groups are sharing abstracts at the upcoming ASH meeting in December. And to cut a long story short, they're not the same. They can have very, very different outcomes. Our research program is focused on BCMA because this is our latest CAR that we're trying to develop for broad clinical use. BCMA has been pretty effective. On the upper right is the 5-year follow-up. The more recent data shows an even perhaps leveled out curve slightly higher, so it's effective. But the most effective one, cilta-cel, also has neurocognitive issues. It's associated with high CRS, high neurotoxicity, and a slightly better remission rate than the competitive product. Nevertheless, this was of concern to us. So we created a whole new set of BCMA CARs. Now, the interesting thing about the cilta-cel-like CAR is it's composed of 2 single-chain binders linked to 4-1BB and zeta. And these were derived from camelids. So you can immunize an alpaca or a llama, and they produce single-chain antibodies. And you can actually put them side by side to double your bang for your buck. So you get 2 binding hits on 1 protein. So we selected 22 different binders for BCMA, screened them for effectiveness, created different combinations, and came up with a final product that we don't have a final product name for. But in specific lytic activities, it's just as strong as our cilta-cel product. This is a specific lysis activity. And when we undergo the mouse trials, where we put in a multiple myeloma cell line that expresses luciferase, we can see that the cilta-cel product and our product, and this is the curves for the individual mice in red and green, they're both highly effective at controlling disease. Now, we noticed something very, very interesting with the cilta-cel product is when the T cells were sitting alone in the tissue culture dish with no tumor there whatsoever, they were still making cytokines. This means that the T cell was self-activating. So you look here in red, this is the cilta-cel-like CAR T cell, which has both of these binders. We said, I wonder why that's happening. Maybe they're interacting. So we split them into separate products, the VHH1 or VHH2 from cilta-cel, and again, comparing it here in green to our own product. And you can see that it's actually this VHH2 domain, just this one alone, is causing the interferon gamma, TNF alpha, and interleukin-2 spontaneous release. So it's self-associating and activating the T cell that carries it on its surface. We looked at the metabolic profile using the Seahorse machine, which measures the oxygen consumption rate and the extracellular acidification rate of these different constructs. Again, it was the cilta-cel-like or just the VHH2 domain that was driving this activation program. And we wondered, hey, I wonder if this actually has an association with the neurotoxicity. So neurotox is really hard to model in the animal system, but we found a cell line, the HMC3 microglia cell line, and we put it on Excelligence and cytokine activity assays. Excelligence assays is a monolayer of this HMC3 cell line. And so as the cell line is growing on this monolayer, if lysis occurs, we lose impedance. It's kept here. So none of the untransduced T cells, our BCMA, and the VHH1 domain are not toxic. However, the cilta-cel and the VHH2 is. This corresponds to cytokine production. And again, I think we've basically demonstrated that the HMC3 microglia cells show that there's a better safety profile when that VHH2 domain is not there. And hopefully our product is going to have a lower tonic signaling and severe metabolic profile once we move on to the clinic. And our poster at the upcoming ASH meeting is going to show which adhesion receptors are responsible for this. So we have a really large translational CAR T cell roadmap, both in hematologic malignancies and in solid tumors. I haven't gone through all of our solid tumor targets, but whether we're looking at starting things or moving them forward into solid tumors, we've been able to move CAR T cell therapy forward on a broad front, both single chain and tandem binders. I want to turn the corner here a little bit and think about other gene vectors and gene delivery platforms, because a lentiviral vector, although close to my heart, is not the only one. In its simplest form, the Pfizer-BioNTech or Moderna COVID vaccine is a gene delivery vehicle. It's a lipid nanoparticle that delivers a messenger RNA that encodes the coronavirus spike 19 COVID spike protein, and thus induces an immune response once that's translated into protein. It's a transient somatic gene therapy, meaning that the vector delivers the mRNA expresses the protein, but as that mRNA degrades within the cell, it's no longer expressed. So it's transient. Now, this sounds easy, but it's actually quite difficult to do because messenger RNA is unstable, especially in vivo. It's highly immunogenic. Your immune system is really poised to identify these, and it's really hard to efficiently deliver. So the reason why Katalin Karikó and Drew Weissman got the Nobel Prize in 2023 is because they came up with the solutions to optimize the nucleic acid structure, putting the right cap motif on the RNA, using pseudouridine, and then figuring out how to encapsulate messenger RNA using ionizable lipids. And so labs across the world now are trying to create their own LNP formulations in order to create their own nucleic acid delivery systems. And small machines exist that easily make these lipid droplets that you can encapsulate message RNA in. The four major types, you need an ionizable lipid, of course, which we just mentioned, which is the key technological insight, some cholesterol, a phospholipid, and then a pegylated lipid. And when these are mixed in the right proportions, we get micelle-like structures, lipid nanoparticles that encapsulate the message RNA. Now, interestingly, we can also chemically conjugate binders or proteins, often using maleimide chemistry, onto the surface of a lipid nanoparticle. So if you add a specific antibody onto the surface of your LNP, you'd have a targeted LNP, a tLNP. For example, if you added an anti-CD117 antibody, your lipid particle will now preferentially target hematopoietic stem cells. And that's really, really exciting because now we're starting to think about how can we start to engineer cells, specifically target them with a chemical composition like vector, giving them at least transient expression of this message RNA. So we're now entering another new era of in vivo cell and gene therapy. And that field is rapidly advancing, especially in vivo CAR T cell technology, which is moving much faster than I would have anticipated, although I've had a little bit of insight. Umoja grew out of Seattle Children's and the Fred Hutch, and they create a recombinant lentiviral vector particle that has on its surface the cocoa glycoprotein for fusion, and then it has a modified binding domain that has 3 different proteins that bind to T cells, giving a great degree of specificity. So this is an example of an engineered lentiviral vector particle. Now, Capstan Therapeutics, it's completely a targeted LNP particle, and in this case here showing you targeting anti-CD117 or c-KIT. This list of companies, which I cut and pasted out of OncoLeader magazine, this keeps growing because it keeps being more and more exciting. And basically every major pharma, whether it was ESO Biotech or Capstan, has created either a deal or completely acquired these in vivo CAR T cell companies. The other major topic that we talk about cell and gene therapy is treatment of the hemoglobinopathies, which is an example of a monogenic gene disorder that we can address and cure using the appropriate interventions. Now, there's actually a number of different ways we can do this. So the current solution that was originally proposed was gene addition. So that's using a lentiviral-based gene expression vector, taking CD34 or bone marrow stem cells out of the bone marrow, and then adding either a fetal globin expression vector or the beta globin expression vector. And this is often mutated so that the amino acids are changed and there's less sickling potential, even if it's incorrectly or the wrong proportion of globins is expressed. We've now moved into the era of gene editing and gene writing, where we can use CRISPR-Cas9 to knock out a transcription factor, BCL11A, that suppresses fetal globin expression. And it could be CRISPR-Cas9, zinc finger nuclease, whatever gene editing technology you choose to use. You can edit BCL11A or other transcriptional regulatories factors that govern fetal globin expression. And we've also now moved into what are called gene writers, that using the same sequence recognition technology that came out through the development of CRISPR-Cas9, we don't just snip the DNA to create a mutation that stops expression of something. We actually can correct the sickle mutation. So there's some gene writing enzymatic capabilities. And the first patient to receive a corrected or gene edited sickle cell locus is here, Brandon Baptiste, December 5th, 2023, standing outside of Boston Children's. So again, this has gone very rapidly from the theoretical to the laboratory, to the model system, and now to being a therapeutic modality. So I'd like to acknowledge a very, very large team. Our team is both in Gaithersburg, Maryland, as well as in Germany. Some of them are in bioindustry, the medical aspects, of course, in biomedicine. And I couldn't have done any of this without them. And I really share with you the advances that we're making with them. So I think that rounds up my presentation. Thank you again for the invitation, and I'll be happy to take any questions or discuss.
Thank you so much, Remus, for this great overview. And we are very grateful. Of course, we have different categories of people on the call, but there are questions. And as you answer them, we will also request you to write written answers so that people can come back and read these to understand better. We have, for the members who are attending, there is an evaluation feedback form. Please fill it before you log off this call. I think it will appear in your window as we go on. I will hand over now to Lois to navigate the questions that are coming up. Very many and very interesting. Some are from me, but I preferred to write them here so that they are documented. Thank you, Remus.
Thank you.
Thank you so much, Dr. Sise. And thank you so much, Dr. Remus, for the wonderful presentation. For the members online, kindly don't forget to fill in the evaluation form at the end of this. I've seen someone asking for answers to the previous session, and I'll kindly remind IT and the coordinator to follow up on the answers to the previous questions from the previous session. Now, our first question is from Moses Supercharger.
But maybe to add in, the questions are uploaded on, as soon as the presenters answer them, they are uploaded on our website. So you can, first of all, access the presentations because we are recording them. And the presenters have allowed to share their slides with us, so they are also available. And the answers and questions are posted on the website when we get them. I think we have the ones from the first webinar. They are already posted. We are expecting the ones from the second webinar. And Remus has had himself. He will respond to these, all of them, so that we can also post them for people to go back and have a more learning experience. Yeah.
Okay, thank you so much, Dr. Sise, for that clarification. So people go to the website and you'll be able to get access to the previous presentations and also to the questions that have been posed before. So our first question for you, Remus, is that CAR T cells are made of 3 engineered antibodies to be called chimeric. Why 3, not 2 or 5?
Well, you skipped Moses Supercharger's question. So
yeah.
greetings to my brother Moses Supercharger. It's been a while. I hope you're doing well. I hope the center is doing well. So chimeric simply refers to different pieces put together. It can be 2, it can be 3, it can be 5. What we're often limited by is the constraints of the gene vector itself. Once we start putting in too many things, we don't get very efficient expression. So the original chimeric antigen receptor, second generation, just has 2 signaling domains and 1 binder, and that's it. It's quite efficiently expressed. As we move on to 2 binders, it's expressed not quite as well, although it's sufficient for high activity. 3 binders, it gets a little bit tricky, and so on and so forth. So we're limited by some means by the vector itself. We can't express an infinite number of things. There's just too much damage and too much stress to the cells. We modify it with those vectors. What we have to prove to the regulatory agencies is that we're not inducing lots of mutations, right? This term is called genotoxicity. And the example I'll give is one of the first CRISPR approaches that was presented for CD19 CARs. If you looked at the genome of those cells, it was quite messed up. There were chromosome translocations, genes were dropped out, lots of lesions. It looked really bad. So these technologies are quite potent, and they have to be very, very carefully analyzed to see actually what they're doing to the genome when we insert them. Now, the gene-based the virus-based ones, like especially the lentiviral-based, we have so much experience with them, and they're kind of based on the biology of the lentivirus. It somehow is almost self-regulated in terms of how many insertions and how much of a gene product we can insert. So we're kind of bound by the size of the original lentiviral genome itself. We can say perhaps Mother Nature has done that work already for us. When we move into the more synthetic systems like CRISPR-Cas9, we got to be really, really careful. If we have the wrong guide RNA, if we overexpress it, we start to get a lot of damage to the genome. You're muted now. Sorry. Okay.
Sorry, thank you, Remus. But also to think about a cell, like although it can express different proteins, different antibodies, but it's also kind of a closed system. So you can imagine, you think about it that way, how much can you introduce without distorting the cell itself, aside from the limitation by the vectors, the cell and the DNA mechanisms. So you have to think about all that when you're designing these CAR-T cells. Your next question is, gene therapy has been globally used to cure cancers and sickle cell disease. Why has it taken long to use it on people with HIV who do not have cancer? In other words, why isn't it within mainstream HIV treatment protocols?
Great question. And it's a debate everywhere. The first patients that were treated with CAR T cells were leukemia or lymphoma patients who had relapsed after 3 or 4 courses of therapy. So basically, they had no hope left. So what was viewed as a highly experimental therapy was seen to at least the internal, the review boards thought, okay, yes, this is a risky thing to introduce gene therapy, but these patients really have no recourse. Now, the more we did it, the more we understood the safety profile of doing CAR T cells. So once we started to understand the safety profile, what are the side effects? What are the best ways to process these? Now we started to think about, okay, what other cases might this be used? The other really impressive use case of a chimeric antigen receptor, specifically targeting CD19, has been an autoimmune disease. We've had amazing results with lupus, as well as other diseases, probably myasthenia gravis, and the universe of autoimmune diseases that are being completely changed by CAR T cell therapy keeps increasing. And I think that's worth a whole nother lecture. But we've spent decades and decades trying to understand how to do something about autoimmunity. And now CD19 CAR T cells work really, really well. Okay, now the question about HIV. HIV, diabetes, lots of what we consider are now quote unquote chronic diseases. Are they well controlled? No, there's still lots of issues, still lots of people dying, still lots of transmission. But we get the argument even here in the US, like, hey, look, there is, effective drug therapy. So why are you doing this? Now, our answer to that, as always, and there's lots of answers to this, is like, well, we haven't tackled this pandemic yet. And so we need cures that can perhaps do this. Maybe the buzzword is in a single shot. If we can provide a therapy that in a single situation not only cures HIV, but perhaps even interrupts transmission, then it's worth it. All the work that it takes to create the right gene vector, the expense of administrating it, et cetera, if we can really break the transmission cycle. So it wasn't tried first because the risk profile was higher, right? We have to take people off of their antiretroviral medications to test an anti-HIV CAR, right? To know if it's working. And so it has to be done in a highly controlled environment. And if there is recrudescence, if the virus starts to grow back, we need to immediately restart their antiviral chemotherapy. So I think it started off because of the risk profile. In cancer, you're allowed to try things a lot faster, a lot riskier. With HIV, CAR T-cell therapy, among other biological therapies, is still not what I would call completely proven. We're still in the very, very early days. I wish I could tell you that I knew, like with lupus, if I gave you a specific CAR, you would be cured. We still have to do that research. We still have to come up with the perfect anti-HIV CAR vector. We'll see if it comes, but we're hopeful.
Okay, Remus, in the same breath, when you talk about whether CAR T cells can be applied to HIV, and we are looking at like real world applications, there is a question here whether there is any age eligibility for therapy initiation, whether it's lupus, whether it's cancer, whether it's HIV. From the studies that have been conducted so far, what has been the main focus for gene therapies in terms of age?
The age of the patient?
Yes, please.
The first publication for CD19 CARs was 3 adults coming out of University of Pennsylvania, Carl June. And then immediately they opened a pediatric trial. So the age, I don't think we're really limited. I think we've gone down to 1 year. For something more chronic like an autoimmune disease, I think you want to see if the disease is controlled by other means. If it's readily controlled by other agents, then it may not be worth the time, risk, and expense. However, if your lupus has moved on to lupus nephritis, where you're having serious kidney damage or other type sequelae, then I think it's time to consider something like CAR T-cell therapy. So it's a combination of age and pathology. This argument, I think, is even more important in hemoglobinopathies. Like, when do we do cell and gene therapy for sickle cell disease? Because we know that sickle cell disease pathology accumulates over time, right? The more infarcts you have, the more pain incidents, the more lung crises, the more it just keeps getting on and on, right? And so usually the candidates who have been come forward and have received gene therapy for sickle cell disease, they've been adults and they've had quite severe disease. And the measure that was used for approval for those was number of hospitalizations over 12 months. So you went from multiple hospitalizations to 0 to 1 over 12 months, which is a dramatic change. The approved products for hemoglobinopathies only went down to age 12. We know, especially sub-Saharan Africa, there's been a lot of damage by age 12. There's been children already dying by that time. So how can we get it to younger kids and to the kids who need it? I think that's really a challenge. I want to join with you in addressing this challenge.
Okay, thank you so much, Remus. There is a question about potential side effects of using lentiviral vectors and what other approaches can be used if one can't use lentiviral vectors. And I'll couple that with the other question, which was, you'll stop moving the questions, because now I can't see. Yes. And can you talk about the non-viral delivery systems like lipid nanoparticles and how they can be optimized to match viral vectors in terms of safety or effectiveness?
Okay, I'll start where you don't expect me to start. I'll start with the AAV, the adeno-associated viral vectors that are used for multiple sclerosis. There were 2 deaths recently this past year with the AAVs for multiple sclerosis. The reason is to reverse multiple sclerosis, you got to put in a lot of viral particles to try and hit all the muscle cells. And that's a liver toxicity. The body can't handle that many viral particles being infused. We see the same thing with lipid nanoparticles. Lipid nanoparticles on their own, we just can't flood the body with a huge amount of lipid nanoparticles and not have damage to the liver or other organ systems. So it's not benign in that context. There is damage that occurs to those cells as the lipids fuse into them, as the nucleic acids are delivered. The body does have some alarming systems that recognize the genome is being messed with. So there is a dose limiting toxicity. AAV, what people are developing right now is trying to make it more specific targeting with less immunogenicity. Lipid nanoparticles would be the same thing. We want to start targeting those lipid nanoparticles. The more specificity you have, the lower toxicity you have. So depending on how many genes you need to modify and how well you target, that'll determine your toxicity profile. It's still pretty early in the era of LNPs. Can we target the hematopoietic with LNPs with a CD117 antibody? We'll see if it's efficient enough. If we get enough targeting to the bone marrow before causing systemic toxicity, especially liver tox. Now, when we talk about ex vivo gene therapy, that's not the same issue, right? Because we take the cells out of the body, we put them into some type of bioreactor device, add our gene vector, wash it away, and put them back in. So if we haven't really damaged those cells too much, they'll be fine once we reinfuse them. With direct in vivo viral vectors, we don't know. It's still really early days. The use case right now, oddly enough, has been mostly for autoimmune disease because it's thought that those patients are somewhat healthier. They can handle more insult compared to a cancer patient. And they only may need transient expression. They may not need to be there for as long and as strong. So it's very, very early days for direct in vivo, but we will see. So again, it's how many cells do I need to modify? How much gene product needs to be delivered? It's always a balance. It's not, there's no free lunch. You can't infuse the body with an infinite amount of particles without toxicity.
And there is a question here about CAR NT cells, whether they are safer and equally as effective as CAR T cells. Which one is more likely to cause cytokine storms and graft versus host response? Any comment on this fairly new option regarding activity and probably lower adverse effects?
This is CAR NK cells?
The person wrote CAR NT cells, but I think it's NK cells.
Yeah, NK, yeah. So NK cells are active field of investigation. They have a different biology than T cells. We've only recently been able to gene modify them effectively now that we have this new envelope, which is why I spent all the time in the beginning talking about the new envelope, the baboon envelope. So now that we can efficiently gene modify, I think we're going to learn a lot more. The current use cases have been after bone marrow transplantation. For example, cells from the donor being given to the recipient. They're supposed to be allogeneic so that you can give as many NK cells to, like NK cells from one donor could be expanded and given to multiple recipients. Now, because they recognize cells not through MHC, they don't cause graft versus host disease, but they are rejected by the immune system sooner or later. So the recipient does recognize that as a foreign cell. So it's not an unlimited amount that you can give as well. So we're still learning how to use CAR NK cells. I think it's still a little early, but it's exciting to figure out how to do it.
Okay, and between ex vivo and in vivo gene delivery, how do you confirm successful delivery to the target site? And in case the gene is delivered or inserted in the wrong site, then what happens? Viral vector delivery is more exposed to mutations. How do you ensure that the processes are stabilized?
So the advantage of ex vivo or even transient ex vivo is that I can sample that cell product. I know what's going back into the patient. So if I gene modify my cells with a viral vector, for example, I can do sequencing and figuring out where did that insertion happen. I can figure out the frequency of those insertions, and I can do some laboratory-based analysis of those cells' functions. So in some sense, we have a better mechanistic understanding. Insertion sites, the safest is lentiviral vector over retroviral vector. Retrovirals insert close to promoters, so they have a higher chance of insertional immunogenesis. That was shown in the transduction of hematopoietic stem cells. No one's ever going to use a retrovirus to transduce a hematopoietic stem cell because we see that that causes leukemias. That's why it's only used in peripheral differentiated T cells. And then lentiviral vectors, it's based on HIV. We know that we don't see many, if any, HIV-induced malignancies, right? Kaposi sarcoma is a different virus, HHV6, and that's brought about because of the immunosuppression. We don't see, although there's millions of infections, we don't see a lot of malignancy that's directly driven by HIV. So we know that the mechanism, the machinery that virus has evolved for gene insertion knows how to pick the right sites and knows how to persist long term, which is why it's an effective gene vector. So we're just piggybacking on something that's already been selected for by that biology, the lentivirus biology that we see in HIV.
Okay, and there's someone here asking for any links to a database for differential gene expression and whether this is pretty accessible. I'm not sure about that because genes are expressed depending on the condition.
Well, if you go to Javed Khan's laboratory at the NCI, it's called the Oncogenomics Lab. And you get a password, you get into his database, and you can do differential gene expression analysis, normal tissue versus cancer tissue. So we've developed this over the last 15 years of working together. So those pediatric databases are easily queryable. I think anyone right now in bioinformatics, it's moving so quickly. Differential gene expression is really, really key. The only asterisk, the only caveat is that mRNA doesn't always equal protein. So someday when the mass spec people get more and more specific, they can tell us, hey, at the protein level, this is expressed on the surface or not. So the databases are there. So dive deep into some data science and some bioinformatics.
Okay, then there's a question about how do we safeguard lentiviral vectors from becoming effective. I think that would be like a case of recombination of some sort, or how can you ensure that they are how do you actually ensure that they are safe and cannot cause any infection?
Yep. Yeah, that's the original, that's again, the original part of the first part of this lecture was how we sequenced the HIV genome and then we split it into 4 plasmids. And then we kept changing those plasmids till we know that we took out all the replicating genes like TAT. We disabled the 3' promoter so that it's now self-inactivating. It's a one-way delivery of that sequence. We know that we need an internal promoter. And the reason why it's 4 plasmids is that the ability for that to recombine, and create an infectious particle is 0. The genes aren't there to reassemble. The fear always was, let's say I'm doing gene therapy on a cell that has an endogenous retroviral or lentiviral infection, maybe one that I don't even know about. Is it possible to bring in some of those elements into an actively replicating virion? So we haven't seen this. The replication rates, it's a set, you know, Mother Nature always likes to make fools of us, right? So, I mean, we can say it's mathematically impossible, but, so far it's impossible. And
Yeah.
we always do a test when we make a lentiviral batch for something that's called RCL, replication competent lentiviral vector. So it's an essential release criteria when we create these vectors that the FDA and the European regulators make us do.
Okay, you've just brought in the aspect of regulators and for example, Uganda, we are just beginning our journey in gene and cell therapy, and there is a question here of what is the lowest standard we can adopt in terms of regulatory aspects and GMP levels for gene therapy manufacturing in low and middle income countries. What is the lowest level? Because you showed us the other farm, which had many prodigies, and then you also showed us a simple laboratory of the gentleman who has treated over 100 patients. So, like, where do we begin from?
Well, I think that's where you begin. I wouldn't call it the lowest level, however, okay? Because I think that's just the wrong word. I would say, I think the safe
requirements, minimum safe requirements.
No, I tell you, I mean, I didn't mention specifically, I was just in Brazil, and Brazil is over-regulated. Everything has to be in a complete clean room. Everything has to be isolated, and this is going to slow them down. Only some specialized centers, and it's in the end, what's going to happen is, I think, unless we work with them properly, introduce the right systems, they're just going to have very expensive therapies that are made outside their country, and that's not the solution anybody wants.
No.
Now, at the national level, what you want is you want the same safety standards. You want the same release criteria. And if you can meet those same standards in a different type of facility, I wouldn't call that the lowest. I would call it the most efficient or the most optimal, right? So let's say I'm only going to treat 300 or 400 patients a year. Maybe that's the disease burden, right? So I don't need to make a commercial factory that can do a few thousand, right? I need to have a trained staff that understands the safety and release criteria and demonstrates that the product is safe. And you demonstrate that that product is safe within the environment that you can manufacture it. So a controlled laboratory environment with clean hoods. I mean, I saw your tuberculosis facility at the JCRC, so I know it's possible.
Yeah. Okay.
Sissy won't let me take that over for some reason, but
you have to talk to her one more time. Then back into making CAR T cells. So there was a question, given that CAR T cells, there are issues like longevity of the cells and also potential exhaustion. There is a question whether we can have them produced by actually engineering stem cells.
Yes, that has been a proposal. On the one hand, I say that's a great idea because we just have to do one modification and it keeps going. On the other hand, I think about, do I need this CAR T cell to be there forever? If it's there for 1 year and my disease is gone, maybe it's okay that it goes away and I get back my normal B cell counts. We're seeing this in the lupus patients for autoimmune disease. The lupus patients get the CD19 CAR, their peripheral B cells go away, and then, I don't know, half a year later, they start to recover their B cell counts, but their disease hasn't returned. we've reset their immune system. So if I can give an effective therapy that removes the leukemia and lymphoma, and then down the road, I think it's okay if the cells are no longer active, if there's no more disease. But it's a very hot topic. I like the question, and you can do a PhD thesis on that question. So.
Okay, I'm going to ask Adrian Wright to talk so that he can pose his question directly. Adrian.
Yes, hi. I hope you can hear me. Perfect.
Yes.
Yeah.
Okay, so first of all, thank you, Dr. Remus. One of the questions, actually I have two questions. The first one is more geared towards the first part of your talk, and then the second towards the second. You mentioned one of the limitations really of the CD19 CAR with respect to, or that approach with respect to solid tumors is that we don't really have markers for the solid tumors. So I'm just wondering, are there groups working on perhaps engineering or in some way trying to manufacture markers on solid tumors that would actually enable it to I guess, be amenable to the same type of approach that you talked about in the first part of your discussion?
Yes. So we are developing different binders that can bind with relative specificity to solid tumors. Some of them have proven to be kind of ineffective because they go to the wrong place and somehow the body tolerizes them. And some of them have proved to be toxic because they target too many normal tissues. I mentioned the GD2 CAR, which targets the disyalloganglioside through a well-developed anti-GD2 antibody that we know a lot about. And it seems to be having an effect in neuroblastoma. We'll see if it happens long-term or not. So it doesn't target CD19, it targets the GD2 molecule, but it seems to be giving us some effects. So I think we're going to try to find this window of specificity in solid tumors. That's really the challenge, Adrian.
Okay. Perfect, thank you. The second question I have, and again, just indulge me here because I'm a little bit spitballing, but I'm trying to wonder, is there a way to actually target the mature red blood cell with any of the techniques that you've mentioned, particularly with with the second part of your talk? I know that obviously red blood cells, there's not a lot in there, but there is carbonic anhydrase as far as the enzyme. Is that a potential way to, for example, in hemoglobinopathies, which, let's say, for example, you have a certain portion of them that still have hemoglobin S or some other undesirable feature, but you have certain cells that you do want to keep. Is there any way that you could actually in the peripheral blood itself target red blood cells that again have been let's say strongly indication of polymerization or already polymerized, or the irreversibly sickled cell? Is there a way to target just specifically those cells and to get them out of the circulation with any of the techniques that you've used here?
It's a great question. I think by the time we hit a mature red cell, it may be too late to intervene at the genetic level.
Right.
Now, I had a conversation 2 years ago with Philip Lowe, who thinks about this. There's lots of kinases that are present and still active that seem to be governing the shape of the erythrocyte, that if we inhibit them, we do get less sickling. I'm not an expert on those kinases, but I know that some kinase inhibitors have proved effective. So I think that's a developing field, and there's more to the biology of the red cell than we know. I agree with that. So we just had a disappointing phase 3 result for another anti-adhesion receptor, right, for the P-glycoprotein. So that hasn't seemed to be panning out quite as well as we hoped. So yeah, there are other interventions that maybe govern the shape of that red cell, and the kinase inhibitors seem to be a reasonable target right now. So.
Okay. Thank you, Remus.
Mm-hmm.
Appreciate it.
Thank you, Remus, for answering our questions. We have about 5 minutes to the end of this session, and there are many questions. These questions will be forwarded to Remus so that he can be able to give more in-depth answers, and they'll be put up on our website. IT team, kindly share the link to the website. Where have you shared? The link has been shared in the chat so that you can find the rest of the questions and the answers and also the presentations. When is our next webinar? 8th. Our next webinar is 8th of September, same time. And Remus, this is a question that is on the hearts of Professor Judy Makani and Dr. Sisi over here, plus other Ugandans that are interested in this cell and gene therapy and moving it off the ground. You've visited us, you've visited Tanzania, you've been to the UK, and you know how you've been to Brazil. You've seen how things are, you've gone through this journey in the US, and now you're in clinical trials and pushing for therapies to go to mainstream treatment protocols. So what would be the 3 strategic things that should be done with regards to establishing lab services for research, especially in the focus of gene and cell therapy, when you look at Uganda and Tanzania? What do you think are those 3 strategic things that we can push for in order to have this cell and gene therapy moving?
Okay, first I put a link to the Oncogenomics lab in the chat. Someone asked about that link, so that's there.
Thank you very much.
Okay, so the key assays that need to be done already can be done. The number one is the number of genomes or number of lentiviral genome copy numbers. So we need to know how many insertion sites we have per genome. That's a PCR-based test. I know that you have great PCR-based activities there, so that's not really a stretch. You just need to know number of copies per genome, like any other gene insertion. So if you can detect HIV levels by PCR, you can detect lentiviral gene modified cells. So that's kind of the molecular biology side. I think you want to have a T cell function side. So I've inserted my gene. Is it active? So I need to be able to take a gene modified cell and show that it does something. The simplest thing is something like a gamma interferon production. Again, that's often a laboratory test that you can do. So if I just put my gene modified cell and something that activates it through that gene, I should be able to demonstrate that, okay, I have an activity matrix, and then flow cytometry to show that the CAR is on the surface of the T cell. So that's the laboratory background we need, right? So copy numbers per genome, that's PCR. Flow cytometry, I got my CAR T cell on the surface, and then some potency or activation assay. For example, I activate my CAR and the cell makes interferon gamma, right? Something that shows. So once you have those 3, the rest are basically kind of microbial sterility tests that all hospitals do. So that's the kind of the basic science setup. So now we have an immunology lab that can do an activity of a T cell, that can look at it by flow cytometry, and that can actually measure some activity. There's other ways you can measure the activity that we can talk about. Once those parameters are set, now we have to think about the therapeutic profile, right? Do I need an apheresis product, or can I start with whole blood? I think we can start with whole blood. There's enough lymphocytes there. The apheresis product is nice because it gives us lots of cells and lots of margin of error. And CAR T cell therapy started with apheresis products because oncology centers do that, right? And but is it necessary? Probably not. But we started doing these therapies. We didn't know how many cells we needed, right? We didn't know how efficient it was going to be. So it's kind of like once that ball started rolling, everybody started using an apheresis product. But clearly now we know for most patients, there's enough T cells in the peripheral blood, so we don't need the apheresis. So that knocks off a big piece of the cost, right? So if we can have a way to isolate T lymphocytes, add the gene vector, and then using the 3 assays I just mentioned, I think we have a CAR T cell product. So is that clear? Is that good?
Yeah, that's good. Remus, do you have some 5 minutes for just 2 more questions? I think this was burning.
Okay, 5.
5 minutes. Okay, thank you so much. So the question is about gene therapy for HIV. Whether there have been any discussions and has it been considered? And then the other question was, I think to tally it up, is whether there are any products in the pipeline for improving strategies for reversing HIV latency. And then we can close it up.
Well, so as you know, the current CAR T cell approaches for HIV that are being tested at UC Davis were developed actually by a team many years ago when Boro Dropulic and myself and Kim Anthony Gonda were all at Lentigen, right? So we are really interested in making sure that those vectors get tested and looking for different ways to go about that. There is a trial supported in California by UC Davis with Steve Deeks and Meredith Betty that have started to test anti-HIV CAR T cells. They have not published any data, so I can't talk about it, right? But it's very early. We don't know if there's a signal or not. But yes, the era of CAR T cell therapy has begun. Once we get that proof of concept, then we can figure out how we can streamline it and make it cheaper. Do we want to start now in Uganda and Tanzania? That would be great.
Okay, thank you so much, Remus, for your time and your patience and your expertise. All questions that have not been answered will be addressed a little later on. You can follow up on the link that has been shared on our website. The next webinar will be on 8th of September at the same time. And yes, thank you for joining us for this very, very, very exciting webinar. Thank you once again. Have a good night. Good morning once again.
Thank you. Okay.
Bye.
Thank you.